Dear Astroglial, many thanks for the nice introduction. Actually, I have to thank you for helping in organizing this symposium. So without the help of Astroglial, Mridula Bala and Hyojin Kim, this meeting would not be possible. And actually, when we are addressing sex-specific differences, first of all, we have to think of the sex chromosomes, the X and the Y chromosome. And I just would like to mention that 900 protein-coding genes are expressed on the X chromosome, while there are only 100 protein-coding genes expressed on the Y chromosome. So on one hand, men appear to be a deletion mutant in comparison to women. When we are looking at all the different genes with their different functions, we will identify that there are many differences in all over the body. So we have differences between the sexes in the heart, in brain function, in the immune system, etc. However, for the current topic, for this session, we will focus on the differences in brain function between female and male animals and between male and female patients. So the outline of this session is the way that I will start to give an introduction. I will highlight some of the unpublished research of my lab, which I think is really gives some insight into the differences between male and female mice, but with relevance to male and female human beings. Then I'm very happy that we have Professor Feng Wang from the Shenzhen Institute of Advanced Technology, since she will tell us some more details about AI-assisted behavioral analysis, looking at different sexes. We will have Yongchang Park talking about transcriptomic data and the respective differences. And finally, Professor Eun-Ja Kim will discuss neurodevelopmental differences. And although on the program it has been written that we will have a question and answer session at the very end, so I think we have a slight change in the program in the sense that questions will be possible after each individual talk. Yeah? And so without further delay, and I think we are perfectly in time since it's written that 10:50 I should start my presentation. So we are 2 minutes ahead of time despite some technical issues we had. So let us start. I will discuss a particular topic. So about astroglial GABAv receptors and GHB-triggered absence seizures. And I will explain this in a little bit more detail. But I had to promise my university president that whenever I give a presentation outside of Germany, I have to explain where we are. So, and as you see here, this is the outline of Germany. Okay, so this is the outline of Germany, and here is Hamburg. So we are basically at the very end of Germany, in the very western part. However, this very western part, we are in the very center of Europe, if you think here. This is Spain, France, Poland, etc. So we are in the center of Europe, just to please remind that where we are. So my own lab, over the last 30 years, 40 years, we studied the interactions of the different cells in the nervous system. On one hand, we discussed how neurons and glial cells communicate to each other. And we are particularly interested in the molecular and cellular mechanisms. So what are the molecules that are sensed by the glial cells or that are released by the neuron to communicate between the different cells and to address brain function. So on one hand, we have the neurons, but then we have the glial cells, which come in different flavors. We have the astrocytes that take up nutrients from the bloodstream and release gliotransmitters, thereby affecting neuronal communication. We have the oligodendrocytes that form the myelin sheath around the axons and ensure fast action potential propagation. And last but not least, we have the microglial cells, the immunocompetent cells of the brain. And for the current topic, I will really focus largely on astrocytes. So those cells that are bridging the gap from the periphery, so from the vasculature, into the neuronal parenchyma. Actually, on this combination of raw data, on this data, you see a synapse. You see here the presynaptic terminals with some presynaptic vesicles, and you see this darker structure. This is actually an astrocyte process that surrounds the synapse. And if you look more carefully at the molecular repertoire of the astrocyte, you see that there are many receptors. There are glutamatergic receptors, GABAergic receptors, purinergic receptors that equip the astrocytes to sense the neurotransmitters released by the presynaptic terminal, but also, getting some retrograde activations. And this is a three-dimensional cartoon of a particular synapse in the cerebellum. And in gray, you see how the glial processes surround this brownish-shaped synaptic structures. In orange, you see the synaptic cleft. But you see how closely astrocytes and neurons interact with each other. And here at this interface, they are equipped with the respective sensing molecules. And we ask the question, how do these glial transmitter receptors contribute to brain function and behavior? However, as a scientist, so we always are divided, or neuroscientists are divided into classes. On one hand, we have the basic researchers that are interested in the molecular and cellular signaling mechanisms, as I am. But on the other hand, we have the clinician scientists that try to translate our findings into the treatment of patients. So, and therefore, animal models of diseases, these are somehow the bridging part between the basic scientists and the clinician scientists. And if you look at the epidemiology, and this is what clinician scientists are doing, they are really looking, how frequent is a disease distributed in a country, how different is a disease distributed over the age range. And in addition, they are also looking how frequently are diseases occurring in the different sexes or genders. And if we look, for example, here, these are German patients, but I think this is very similar all over the world. We have dementia, and basically at the age of 80 years, we see a clear separation that women are more frequently affected than men. And interestingly, this might even preceded by the differences in depression. So I think we have a certain correlate that depressive disorders start earlier than dementia, and we have to address this difference. However, there are not only diseases or neurological diseases that occur at the end of our life, and however, there are other diseases that are present throughout whole lifespan, like multiple sclerosis, this autoimmune-related disease. This affects young individuals already at the age of 20 years, and you can live with this disease for almost your whole life and become even 80 years old. However, there is a very clear sex-dependent distribution. So we have three times more frequently women are affected than men. So in orange, we see two different years in Germany, and here also these two different in blue, the distribution, how frequently multiple sclerosis is occurring in men. However, despite the fact that women are affected three times more frequently than men, men have the worst outcomes. So men are more frequently wheelchair bound. And actually, this difference cannot that easily be explained by the different levels in estrogen and testosterone. So obviously, we have to look more carefully in understanding these diseases in understanding the mechanisms and then finding respective therapies. And if you look at many neurological diseases, what you find is that Alzheimer's, depression, anxiety is more prevalent in women. Autism, Tourette syndrome, Parkinson's disease is more prevalent in men. So basically all neurological diseases have a certain type of gender-specific distribution. And I'm just, please, when I'm talking about sex, I mean the biological sex as it can be identified. And if I'm saying gender, so with a mouse, I only know the sex of the mouse, whether we have a male or a female mouse. However, if I do not do a physical inspection, I can only read you as a woman or I can read you as a man. So then we are using the term gender. So if we do not know by inspection, by biological analysis, then we're using the term gender. Yeah. And this is more prevalent. When we are talking about patients, we have to talk about gender. If we are talking about mice, we have to talk about sex. However, now, discussing brain functions, so structural differences occur between women and men. And we have now powerful techniques like magnetic resonance tomography or diffusion tensor imaging. And if we apply this non-invasive techniques, we see significant differences, for example, by looking at gray matter volume. And this has been done by Armin Rasnahan. He looked at thousands of patients, male or female, and he identified dozens of different gray matter areas where either men have a larger gray matter volume or women have a larger gray matter volume, or the other way around. So basically, it's not the topic, do men have a larger brain or women have, it is the topic that we are different in different areas of the brain. Yeah? And this is a sex-specific difference. But what I personally find much more exciting, rather than looking at the size differences in different brain regions, this is about the functional connectome. And this is a work done by Ragini Verma. She did a very interesting analysis. She looked at brain regions that were functionally connected. And what you then find is that in the male population, the functional interaction occurs more within the same hemisphere, while women are more connected between the hemispheres. So, and if I told this work, or if I reported this work to my wife, she said, well, this is very clear. Women are much more multitasking able, they can do multitask at the same time, while men are only, focused on one issue. I don't know whether this is the explanation for this data. What you see on the right, these are two different pairs. So on the left, you see a boy, on the right, a girl, a boy, a girl, a boy, a girl. However, this is before puberty, during puberty, and after puberty. And already at the level of individual young human, you see that probably around puberty, we see this separation, that we see more connections between the hemispheres occurring. So therefore, it will be interesting to see whether this kind of functional connectivity has this something to do with the exposure to sex hormones during puberty, or is this inherently regulated? Is this genetically determined by sex hormone independent mechanism, or has this something to do with the education that girls and boys might experience different, might have different experiences during adolescence? So all this has to be addressed, and I think this is the reason why people get more and more interested in looking at the transcriptomics of different brain cells. And so far, in particular, the transcriptomics of microglia cells have been investigated, and very interesting data have been identified, like here by the group of Adriano Maggi. So the group did a nice experiment. They were using male mice, and these male mice were subjected to a middle cerebral artery occlusion, or they were subjected to a stroke. And then either from male mice or from female mice, microglial cells, these immunocompetent cells of the nervous system, were purified. So from healthy mice. So and then male microglial cells were transplanted into the diseased area of a male mouse. And actually what happened was that the injury site, the stroke site, got bigger. But if you transplant female microglia into the male brain, what you find is the lesion gets smaller. So obviously, female microglia cells have to appear to exert a neuroprotective function to the male mouse brain. Unfortunately, they didn't do the experiment the other way around by looking at female mice with a stroke. But anyway, so others now look at autism spectrum disorders, and they identify neuroligin as an important molecule, or they do more other transcriptional analysis. So definitely also at the transcriptional level, we see significant differences even in a cell type specific manner. So we see differences not only in microglia, but also in astrocytes, in oligodendrocytes and neurons. So coming now to my own work. So in our lab, we are interested in a series of diseases. So in multiple sclerosis, but also in epilepsy. And one form of epilepsy, these are absence seizures. Or absence seizures is a type, or absence epilepsy. This is a type of epilepsy that frequently occurs among children. And here you see this girl, it is reading at school. However, then these absence seizures are characterized by generalized non-convulsive seizures, and they have a certain pattern of 2.5 to 4 Hertz spike and fade discharges. And if these discharges occur in the brain, then the patient becomes unconscious and basically appears to daydream and that. And if then these seizures disappear after a few minutes, the subject reawakes, so to speak. And these are, as is written here, brief and sudden impaired phases of consciousness. And we have a complex mechanism which affects a cortico-thalamo-cortical network. So the different cellular components have been identified. However, the modulation of this system is still unclear. However, there's an animal model for these absence seizures. And this animal model is characterized, or is using gamma hydroxybutyric acid, GHB. GHB has become bad, has a bad fame. It is used as a raping drug, or KO drops, since it's almost tasteless and it can be easily put into some drinks. However, GHB is also an endogenous molecule in the brain. It is an endogenous GABA precursor. It does so. GHB can synthesize also GABA, and it is a weak agonist of the GABAb receptor. And so, as it's written here, so you have a KD of about 50 to 500 micromolar. And actually, if you inject GHB, IP intraperitoneally into a mouse, you can elicit spike and wave discharges at a frequency of 3 to 6 Hertz, and this in a dose-dependent manner. And so whenever you have these discharges, the mouse moves, and according to the discharge, the mouse arrests. Now, GABAb receptors belong to the most abundant metabotropic receptors in the brain. They are expressed on neurons, on thalamocortical neurons, on corticothalamic neurons, on interneurons, but they are also expressed on astrocytes. And we were particularly interested in the role of GABAB receptors on astrocytes. So we generated a mouse model where we used an astrocyte-specific promoter to drive the tamoxifen-sensitive Cre-DNA variant Cre-ERT2. So basically using the Cre-LoxP system. We are using a knock-in mouse where the Cre-ERT2 is knocked into the glutamate aspartate transporter. And then we are using a mouse in which the GABAB receptor subunit GB1 is floxed with respective LoxP sites. So, and then these are the control data, you know, that the injection of tamoxifen elicits a significant decrease in mRNA for GABAB receptors, impurified astrocytes. You can do this also with in situ hybridization or with Western blot of max sorted astrocyte. So basically this is a clear indication. We have expression and we can delete the GABAB receptors. And then if we inject gamma hydroxybutyric acid, what we observe is that not all the brain frequency are equally affected. What we see is that the delta waves, and the delta waves, these are the waves between 2 and 6 hertz, they are particularly reduced. You see here under control conditions, we have the activity of the delta waves, and they are reduced in the mutant situation. Well, this is then also quantified here. You see this is the delta wave peak, dose dependently increased, and we reach here a spectrum power amplitude of about 400, while in the mutant we only reach about 200. This is another way to plot it. So this is the power of the presence of these delta waves, and you see these are the control mice and these are the knockout mice, and you see that only the delta power is affected, while the theta power, the gamma power, is much less affected. So it's really, you know, a frequency-dependent modulation of brain activity by knocking out the astroglial GABAB receptors. And what you also see is that the animal activity can be reduced by GHB. So you have a certain activity, and if you inject GHB, the mice become immobile, and this immobilization is less effective in the mutant mice. This is another display of the data. So here we are looking at the different frequency, delta, theta, alpha, beta, gamma. What you see is that here, this is the delta power significantly, and then the other powers are a little bit less. But you see that in the mutant mice, these delta power is significantly reduced in the mutant mice. So now, what we did is we separated the data. So these data here are from individual mice. So now we looked at the sex-specific distribution. And what you see here, that we are looking at the delta power of female mice, and here we are looking at the delta power of male mice. And we were quite astonished to see that the female control mice are much more sensitive to the gamma hydroxybutyric acid than the male control mice, and that this difference could be significantly altered by knocking out the astrocyte-specific GABAB receptor expression. And you see that the activity, this is some other, it's also affected, but here you see there the differences about the activity is not that clear, but here you see at least the delta power clearly reduced. And the other frequency, like the delta frequency, is not affected. Okay, so now, then we looked also at the time span the spike and wave discharges last, and you see a longer wavelength, you know, in the control mice. You have a long spike and wave discharge. You knock it out, it's reduced. And again, if we are separating the data, these are the data of all the sexes. And here we are looking just at the female mice and here at the male mice. You see that the female mice spend larger times, they are affected longer in the condition of spike and wave discharges. We also have, this is the individual spike and wave duration, and this is the total time in the spike and wave discharges, and this is the sum. These are the number of incidents about how many spike and wave discharges you have in a dose-dependent manner. Here, this is the time in spike and wave discharge, and this is the individual duration. And you see that the female mice are much more affected than the male mice, and that also the differences can be eliminated more clearly by knocking out the astroglial GABAb receptor. Then we looked also at the behavior. So we did this open field data, and these are the mixed data where in black you see the control mice, and in blue the mutant mice. So you can reduce the activity by knocking, you can increase the activity with a knockout. And then again, if you separate the data, you see that the differences between control and mutant mice are larger in the female mice. Like here, this is the cumulative immobility. So this means the female mice stay longer as in immobile version under control conditions, so with GHB. But then if you knock out the astroglial GABAb receptors, they are less immobile. So you can really delete the sensitivity to GHB, or this sex-specific sensitivity to GHB, by deleting the astroglial GABAb receptor. Then we looked also at the neuronal activity by looking how differently the neurons might become activated. And one sign of neuronal activation is the expression pattern of C-FOS. And again, you see the GHB actually in this is very interesting. If you are looking at CHB, you see an increase in neuronal C-FOS expression if you inject GHB into the mice. And this increase can be reduced by knocking out the astroglial GABAb receptor. And again, the differences are larger in the female mice rather than in the male mice. Yeah, so we have obviously the astroglial GABAb receptor. I will repeat it a few more times so that one of the take-home message. So we clearly have identified a mechanism that affects brain function in an astrocyte-specific manner. We performed transcriptomic data analysis on mice treated with GHB or not, and having the GABAb receptor or not. And in particularly, we were interested to identifying a gene set which has something to do with adenylate cyclase activity as well as calcium binding. And then indeed, if we performed calcium imaging in acutely isolated brain slices, or in this case, it's even done in living mice. So we are looking in living mice into the brain, and we see this astroglial calcium activity. And this astroglial calcium activity is modulated in the mutant mice. So we have very fast, and so these hypersynchronous calcium waves, which are then reduced, and here are some quantification. So the GHB induces these calcium transients, and again in the mutant mice this is reduced, and also this calcium activity is larger or stronger reduced if you are looking in female mice rather than in male mice. So also, at this physiological level, we can see sex-specific differences by looking at astrocyte physiology. This is just another quantification of our calcium signaling data. So where in the mutant mice, we see a reduction in the amplitude of the calcium signal. And this is in a dose-dependent manner, etc. So here you have coincident activity in the mice expressing the GABAB receptor, but then all this is deleted, reduced in the knockout version. Then we also did we looked at the transmitters which are expressed or which are active in the brain, and for that purpose we used special canules. So we measured the extracellular levels of transmitters, and actually when we inject gamma hydroxybutyric acid, we see a particular release of glutamate. We still have to do a little bit more experiments, but again, this release of glutamate is particularly prevalent. This release of glutamate, well, let's say the release of glutamate is less pronounced in the control mice, but rather in the mutant mice, we see this strong release of glutamate. So this might be that, in this context, we inject GHB. GHB will also affect not only the astroglial GABAB receptors, but it will also affect the neuronal GABAB receptors. So therefore, we think that we have a complex interplay, that the GHB activates, on one hand, the neuronal GABAB receptors, and this then leads in the knockout situation to a particular increase in extracellular glutamate, while in the presence of the astroglial GABAB receptor, this glutamatergic release from the neurons is under control. Yeah, so we have a feedback mechanism from the astrocytes to the neurons. And we can also elicit, when we are using a glutamate sniffer, in cultured astrocytes, we can also elicit a glutamate release with GHB. And this can be reduced when we block the GABAB receptor with a respective GABAB receptor antagonist. So the interesting point now is all this was reported in mice, right? However, if you look at the literature about GHB, GHB is used, as I said, as a raping drug to immobilize women. However, in men, GHB is also, or it has been used by men as a sexual stimulation drug. So obviously, GHB also in humans affect our brain differently. Yeah, so on one hand, it is used for stimulation, and on the other hand, on immobilization. Yeah, so therefore, however, now, these data, which are completely unrelated and from human studies. So however, what we basic scientists are now doing, we try to explain human behavior at the mouse level so that we can feed back and to understand better how we humans are working. And actually, I already showed this slide. Brain-specific differences, well, sex-specific differences do not only occur in the brain, they also occur throughout the body, and in particular between organ-organ communication, the immune system, the nervous system, the heart, etc. Also the interaction between the heart and the kidney. So therefore, at my university, we decided to set up a center for gender-specific biology and medicine, where principal investigators from all disciplines, from cardiology, immunology, nephrology, sports sciences, and basic scientists, pharmacology, physiology, biochemistry, join forces, and we have individual research projects in addressing sex-specific differences throughout the body, either in patients or in our respective animal models. And here you see that the crowd of scientists all of them are laughing. So obviously it's fun to make research. And it's even more fun also to make sex-specific research. And actually I try to be an ambassador of these and try to raise awareness that we have to address sex-specific differences And therefore, we do not only have a Center for Gender-Specific Biology and Medicine at CGBM in Germany, but I founded 3 other CGBMs in Nanjing, in Suzhou, and in Chengdu at respective Chinese universities. We have already one paper from the Brazilian Center of Gender-Specific Biology and Medicine, and I'm in discussion with colleagues in Canada and in Japan to set up one as well. And we are also discussing to set up such a network of PIs that address these important research questions here in Korea. And with that, I come to an end. You see, my group is very active, so if you join my group, it is obligatory to participate in the city run. Yeah, and you see, obviously, although it is a must for the people in my lab, yeah, we have fun to do that. So we have fun to do research and fun to do science and to do cardiovascular exercise. Thank you very much.